everyone. Welcome back to cell biology class. We were just talking about how tumor cells are able to trigger angiogenesis. They're able to cause blood vessels to grow into their tissues. That allows the tumor to get nutrients. It allows the tumor access to blood vessels, to metastasis. As that tumor is going through the bloodstream, those metastases will preferentially develop at certain body sites. These could be sites that give an optimal growth environment. Different cancers will have different preferences. Cancer cells are also most likely to become lodged in capillaries. For a lot of tumors, the first time they come to a capillary bed is going to come to the lung capillaries. But for others, things like stomach and colon cancer, they'll be found in the liver capillary bed. Another thing that travels through your blood and lymphatic system, though, are your white blood cells involved in your immune system. And they also have a role to play in cancer. The immune surveillance theory is postulating that immune destruction of cancer cells is common, that there's all the time little mutations happening, little cancers starting to form, but your immune system comes and destroys them and keeps it in check before you even have any idea that anything happened. So cancer, when it develops, would be a failure of the immune system. The immune response just didn't get to that cell in time. There are some things that support this theory. So transplant patients who, in order to not reject their transplant, are on immunosuppressive drugs. Those immunosuppressive drugs are suppressing their immune system. With that suppressed immune system, they are more likely to get cancers. They have higher cancer rates than normal. Mutant mice, we talked about our mutant mice without an immune response, especially if they have mutations in their RAD2 gene, they have higher rates of cancer. AIDS patients, which is a disease of the immune system, they have higher rates of cancer, but only certain types of cancers. So this suggests that maybe your immune system is involved mainly in protecting us from virus-induced cancers more than it protects us from other reasons that we have cancer. However, cancers are sometimes also able to avoid the immune system. They will do little tricks to stop themselves from being destroyed by the immune system. One thing would be that your immune system learns to respond to particular antigens. So different cells in the tumor might have different antigens. So they'll have different levels of being triggered by that immune response. And as that tumor grows, some of those cells that have a weaker response from the immune system are going to survive, and that's what's going to grow. Those cells that are more likely to be killed by the immune system aren't left to grow. So as tumors grow, they are gradually, that survival of the fittest means your tumor is going to become less responsive to the immune response. Some cancers are going to do things like kill T cells or inhibit T cells. They just make molecules that damage and impair those cells. They might surround themselves with a tissue that just gives them a little bit of a shield from the immune system, so the immune system cannot physically get to the tumor to kill it. Some tumors are growing so quickly that the immune system just can't keep up. As hard as the immune system is working, it just can't kill the cells as fast as they're being created. Cancer cells also are commonly caused by errors in DNA, especially if you have problems in DNA replication and DNA repair, which are going to allow errors to accumulate and be passed on. Environmental agents and lifestyle factors can trigger mutations in DNA, and heredity. Inheriting a bad or a faulty copy of a gene can make you more likely to get cancer. So cancer is a genetic disease, a disease where the genes have been mutated. And the cells that acquire those mutations are going to be the ones that progress towards proliferation and towards metastasis. We can find a lot about what causes cancer by studying epidemiology. This is the study of different lifestyle factors that people have, different habits, different chemicals they're exposed to, what goes on in different people's days, and then studying their rates of different diseases. So one example is that epidemiology taught us about the connection between cancer and cigarette smoking. So for instance, this graph shows you how many cigarettes a person smokes per day. And as they smoke more and more per day, you see taller and taller bars. You're getting more and more chances of developing cancer. How young they were when they started smoking. So if they never started smoking compared to the younger and younger they are, the more likely they are to start cancer. How long it's been since they quit smoking. So the longer it's been, the less likely you are to develop cancer. Almost back to a baseline level here. We've also been able to see different cancers that are linked to smoking. So smoking increases the risk of lung cancer by a huge amount, but it also can increase the rates of other cancers, especially cancers associated with the digestive and reproductive system and not just your respiratory system. So we see the esophagus, stomach cancers, pancreatic cancers, uterine cervical cancers, kidney cancers, bladder cancers, and colon cancers. All of these are increased by cigarette smoking. Another thing that can cause DNA damage is just random failures in DNA replication and DNA repair machinery. This is not caused by their lifestyle. It's not caused by something they're doing. They are just random mutations that happen here. There is a correlation, however, between the incidence of cancer and how many times that stem cell has divided, the number of divisions it's gone through. So older stem cells are more likely to have these errors in DNA replication and repair. There's also this additive effect between these innate mutations not caused by anything, mutagens, so environmental agents, things like things like cigarette smoke that can cause tumors, and conditions like cell stress. So even if you can't control some things, like you can't control how old your cells are, you can control some things, and everything that you're able to address is going to lead to a reduced risk of tumors. The list of known and suspected carcinogens, or things that cause cancer, have grown to have hundreds of different chemicals on them. Some carcinogens are going to need to be metabolically active before they're able to cause cancer. So that means that in their original form, it's not particularly dangerous, but as it gets into a human body, goes through human metabolism, it can be converted into other molecules. That's called carcinogen activation. One example would be this chemical 2-naphthylamine, which just out in the wild is not particularly dangerous, but when it gets into a human body, it can be metabolized into other chemicals. The liver can mutate that into other chemicals. Those other chemicals are what's going to cause cancer. So even being exposed to that precarcinogen, once it gets into your body, it's a carcinogen all the same. Bruce Ames developed a lab test to be able to measure if something is mutagenic or not. His test is called the Ames test, and it uses bacteria that are able to not make histidine on their own. So these bacteria, if you grow them on a plate that has histidine, they can use the histidine that's there. They can grow normally. If you grow them on a plate that doesn't have histidine, they can't grow. They are not able to make histidine. They're not able to eat histidine. They have no access to any sort of histidine, so they're not able to grow. However, if there is something that is mutagenic, that triggers mutations in that bacterial DNA, it could mutate that bacteria to be able to start growing on histidine. So you're able to grow this chemical. This chemical is added to your bacteria. You plate them onto 2 plates, one without the chemical and one with the chemical. Without the chemical, you might have a couple of random spontaneous mutations and a little bit of growth. If your chemical is not mutagenic, you would expect to see this lower rate of cell growth. If it is mutagenic, if it causes lots of damage in the DNA, it's more likely you'll start having cells growing on the histidine deficient plate, and you see all these dots. The only way those dots could be there is because their DNA mutated. So you know that something mutated it. You know that this substance is a carcinogen. Some chemicals, as we talked about, have to be activated, metabolically activated. So you can take your suspected mutagen and mix it with a liver homogenate. So it's incubated with those liver extracts that can activate any sort of precarcinogens. Mix that up with your cells and have the same experiment. We see, are they growing only at baseline levels, or is there a lot of growth, indicating a lot of DNA damage? Carcinogens are able to change DNA in a few different ways. One is they can cross-link the two strands of the double helix. That's going to create problems when the two strands need to separate. Problems with cell replication, with DNA replication, problems with transcribing that gene. They could create chemical links between adjacent bases. So bases on the same strand should be base pairing with bases on the opposite strand. Instead, they're suddenly base pairing with each other. Hydroxylating or removing bases, changing the DNA, temporarily or permanently changing that code that it's carrying, and causing breaks in one or both strands, which means you may have broken a gene, turned off a gene, could have altered the promoter region of a gene. It could have now end up with two different chromosomes stuck together with new genes being created. One gene that we see being affected a lot is P53. Cigarette smoke those chemicals are able to bind to regions of the P53 gene. So this is a gene we're going to come back and talk a lot about because mutations of P53 play a key role in many kinds of cancer. Radiation can also lead to DNA damage. X-rays can create DNA mutations and lead to cancer, and this is in direct proportion to the dose administered. So something like if you are getting one X-ray a year at the dentist, that's safe. They'll probably give you like a lead apron to put on to protect other parts of your body, but you're not likely to get cancer from that. However, the dental hygienist who's doing these X-rays all day, every day, is being exposed to a lot more X-rays. So she's going to step out of the room to avoid having that higher level of X-ray because a higher dose can make it more likely to get cancer. There's also this paint that was used to paint watch dials, and they added radium to it. Radium is kind of really radioactive. It was great on the watches because it let the watches be luminescent. They could see their times better. But the painters, these women who were painting the watches, they'll dip their paintbrush in the paint on the watch, lick their paintbrush to kind of get it back to that nice point, dip it back in the paint their watch, lick the paintbrush back on their watch. Every time they licked their paintbrush, they were getting some radium ingested into their bodies. And so they ended up with higher levels of bone cancer. X-rays and these radioactive molecules create what's called ionizing radiation. And it's called that because when it interacts with molecules in your cell, it'll strip them of their electrons, which leaves them with a charge, so they're ionic. Molecules don't like to have their electrons taken away from them. They get really reactive in a process to get those electrons back. Those really reactive ions are going to generate a lot of DNA damage. So the ionizing radiation strips electrons from molecules. Those molecules become reactive. They get angry and they go try to steal their missing parts from DNA. DNA gets damaged in the process. Ultraviolet radiation is also able to cause cancer. Ultraviolet radiation is mainly associated with like sunlight, UVA, UVB, you've heard of that. And it's mainly absorbed by the skin. So mainly we think of sunscreen as protecting you from skin cancers. What UV radiation does is different from what the ionizing radiation does. What UV radiation does is trigger pyrimidine dimers. That is when you have one strand of DNA that has a couple of thymines next to each other. And those thymines should be base pairing with adenines on the opposite strand. Instead, they start pairing with each other. That is damage that can be corrected by the photolysases in your cell. But if it's not repaired, it can cause problems in DNA replication. So UV radiation causing this pyrimidine dimer not getting repaired is going to lead to tumors. There's also microbes that are able to trigger cancer development. So in the early 1900s, Peyton Roos was doing these experiments on chickens that had levels of cancers of their connective tissues or sarcomas. You may have heard of Roos sarcoma virus at some point. If you haven't, there's something to look up. And so he showed that their sarcomas were being caused by a virus. This was called an oncogenic virus. So onco, like oncology, means cancer. Genic, like generate or genesis, means causing something. So this is a virus that can cause cancer. Another virus that can cause cancer is Burkitt's lymphoma, which is associated with this virus, EBV, Epstein-Barr virus. So Epstein-Barr virus is another oncogenic virus. This is a virus related to herpes viruses. Often these Epstein-Barr viruses, the DNA and the proteins from the virus can be found in tumor cells of a patient with Burkitt's lymphoma. But if you look at the normal cells of those patients, you won't find those DNA and proteins. And if you're growing lymphocytes in cell culture, if you add EBV to it, that can cause those cells, even in culture, to acquire properties of cancers. So EBV is another oncogenic virus. Hepatitis B and hepatitis C viruses can trigger liver cancers. Human T-cell lymphotropic virus 1 or HTLV1 can cause adult T-cell lymphoma. And human papillomavirus or HPV is associated with uterine cervical cancer. These are some things you may have heard of hepatitis, you may have heard of HPV. Helicobacter pylori, this one is different. It's not a virus. Helicobacter pylori is a bacteria. It's often found in stomachs that have stomach ulcers. So it's a cause of stomach ulcers and is also linked to stomach cancer. And then flatworm infections, these would be worms that are able to grow within your reproductive tract. Within your digestive tract. If you have eaten something like some meat that's not been cooked properly, you can get the flatworm infection from your food, and then it starts growing in your digestive tract. These are linked to a number of bladder and bile duct cancers. So knowing these things can cause cancer is important because it allows us to create prevention strategies. Things like creating antibiotics against bacteria like H. pylori, creating vaccinations against viruses like HPV or hepatitis B allow us to prevent cancers. Okay, so that's a lot of things that cause cancers. Next, we're going to talk about the genes in our body, our oncogenes, which are genes that are turned on in cancers, and tumor suppressor genes, which are genes that are turned off in cancers. I'll see you in our next video to talk about these topics. Bye.